Robin Lovell-Badge. I'm a senior group leader at the Francis Crick Institute in London. I work on embryology, stem cells and genetics. Genes have two parts to them, of course. They have the part that encodes the protein, which is functional and does things, and then they have a part which is the regulatory region. Regulatory regions usually have these sequences that are referred to as enhancer sequences, which are required to activate the expression activity of the gene. Now, we were particularly interested in the role of this specific gene called SOX9, which has a very central, early, important role in sex determination. So whether you become male or female, which relates to whether you make testes or ovaries. And SOX9 is essential to make testes. Turns out, though, that the regulatory region of SOX9 is extremely long and complex. It's a gene that has roles in many other aspects of embryo development, actually, and also in stem cells in the adult animal. So we used a variety of methods to try and ask what sequences within this huge region were likely to be regulatory in some way, enhancers sequences. And we found initially about 35 potential regulatory regions. We then just used a variety of methods to whittle them down to a set which was about 16, which we felt were much stronger candidates. And of the 16 we tested, 4 gave activity in gonads. And when we then used genome editing methods to delete each of those 4 enhancers, 3 of them had no noticeable effect at all, but 1 of them turned out to be absolutely essential. So we delete this enhancer, you get essentially almost no expression of SOX9 at all, so that an XY embryo, which of course should make testes and develop as a male, no longer does, and it makes ovaries and develops as a female. We think it's so important because it's the first one that's needed. It acts very early, and not only does it act early, it has to act early to allow SOX9 to be expressed sufficient levels to give you testis development.